In this segment of our video seminar series, I'm going to survey the various ways in which mitochondrially generated heat may be of material importance in diverse cellular processes. Much of what I'll have to say in the next 15 or 20 minutes will be very speculative. Some of it will undoubtedly prove erroneous, whereas other parts may turn out to be on the mark. Time will tell. Last time we considered various findings from my own lab's past work on Drosophila that indicates a biologically material importance of mitochondrial heat production. We looked, for example, at spermatogenesis, midline closure during development, recovery from cold-induced paralysis, and alleviation of proteotoxic stress. We also mentioned in passing many other phenomena, some of which we'll now examine in greater detail in this segment, as well as in parts 6 and 7. Here, we will look at some specific examples from cell biology, including mitochondrial dynamics, motility, apoptosis, and immune cell functions, concluding with a discussion on the nature of heat sensors in mitochondria and in the cell more generally. We're now reaching the second half of this seminar series, where the focus shifts away from experimental findings to mechanistic speculation. As I've already indicated, undoubtedly much of what I will have to present here will prove to be wrong in detail, but so long as you bear this in mind, I think it's important to consider the issues. Importantly, if heat is produced, we have to take account of it, even if it proves not to be instrumental to a specific process. There is already an enormous literature on mitochondrial dynamics, the set of processes by which mitochondria grow, fuse, fragment or divide, and move within the cell. In fact, there are numerous videos freely available on the internet that visualize these processes. Most studies have focused either on the molecular machinery of fusion, fission, and motility, or on the tendency of mitochondria to fuse or fragment suddenly in response to a cellular stress, which has been interpreted as being part of a cycle of changes that facilitate mitochondrial quality control, involving isolating and discarding non-functional parts of the cellular mitochondrial network. However, the literature remains confusing and in some ways contradictory. Here, I'm introducing a new concept that I believe may explain away some of these contradictions, although I'm not proposing this as a new universal paradigm to explain away everything in mitochondrial dynamics, and in fact it might even confuse the picture further. The basic idea is that the intracellular organization of the mitochondria is determined, at least in part, by the rate of mitochondrial heat production, and is a homeostatic process that keeps mitochondrial temperature within safe or optimal limits, and also facilitates or regulates heat transfer to the rest of the cell and beyond, depending on physiological signals. The fragmentation of mitochondria should greatly increase their ratio of surface area to volume, so it's predicted to be a homeostatic response to mitochondrial overheating, or to the cytosol becoming too cold, whereas mitochondrial fusion should do the opposite. In line with this, treatment of cells with an uncoupler, such as FCCP, that should produce a large increase in heat production, results systematically in mitochondrial fragmentation. Treatment with metformin, which has been shown to inhibit electron flow through complex 1, conversely induces hyperfusion. There are a number of other observations in the literature that fit this paradigm. The acquired ability of zebrafish to withstand high temperatures in the environment is associated with mitochondrial fragmentation, at least in the liver. This is consistent also with our idea floated in part 3 of this series that mitochondria somehow prioritize taking care of themselves. By delivering more heat to the rest of the cell, mitochondrial fragmentation would counteract the thermal stress on the mitochondria themselves. And this would occur even at the expense of a temperature increase in the rest of the cell. Similarly, mitochondrial fission was observed to accompany the thermogenic burst in mammalian brown adipocytes when they are stimulated to undergo the uncoupling of respiration and ATP production. The thermogenic cells of plants are also generally filled with small, rounded mitochondria. The availability of tools for profiling temperature in different cellular compartments, as described in part 2 of this series, should assist in addressing if such a relationship holds. Mitochondrial motility has been most studied in axons, where the prevailing view has been that mitochondria move in an anterograde direction, i.e. towards the neuronal synapse, where they deliver ATP, metabolites, and other materials supporting signal transmission to neighboring cells. Conversely, mitochondria return to the cell body when depleted of functional materials in order to be replenished or turned over. However, the possibility that what they are delivering is, or at least includes, heat has been overlooked. Temperature regulation in the synapse is likely to be crucial for the release and reuptake of neurotransmitters. On this view, mitochondria would move in the retrograde direction when they have become permanently cold and no longer able to supply the needed heat. Once more, this hypothesis may or may not hold up, but definitely deserves to be tested experimentally. Mitochondrial temperature may not itself be of functional significance, but may be a physiological marker indicating which mitochondria should be transported and in which direction. I'll now turn to the phenomenon of programmed cell death, or apoptosis in its commonest form, though of course there are many variants. The mitochondrial dimension in apoptosis was for quite a long time considered as a rather minor contributor, with the activation of death receptors at the cell surface thought to be the main trigger. However, the balance of opinion has shifted, with mitochondria now recognized to have a crucial role in the life-death decisions of cells. The outer mitochondrial membrane acts as a key platform for the initiation of apoptosis via the balance of pro-apoptotic and anti-apoptotic members of the BCL2 family, with loss of outer membrane integrity and massive release of cytochrome C from the intermembrane space, effectively rendering the process irreversible, and therefore also self-propagating. Note, however, that in this initial phase, the inner mitochondrial membrane remains intact, polarized, and able to continue producing both heat and ATP. In most animals, this process proceeds with the involvement of an adapter protein, APAF1, which forms an oligomeric complex with the released cytochrome C, plus a crucial initiator caspase, caspase 9, which is proximity activated. This creates a cascade which then activates effector caspases such as caspase 3, effectively dismantling the cell by proteolysis and recycling its components in an organized and orderly fashion. However, I'd like here to propose an alternative concept for which there is currently only really flimsy circumstantial evidence, but which fits the idea of mitochondria as heat-producing engines. This hypothesis is built upon the observation that plants do not have APAF1, nor do they have orthologs of the metazoan caspases. However, they do have distantly related metacaspases that have also been implicated in programmed cell death. Crucially, plants do show release of cytochrome C from mitochondria when programmed cell death initiates. Given this observation, I hypothesize that cytochrome C release itself is an ancient or the ancient mediator of cell death, whereas the caspase cascade, activated by its binding to APAF1, is not ancestral. Some other aspect of cytochrome C's biochemistry is therefore implicated, with the APAF1 caspase 9 switch perhaps playing an accessory or confirmatory role in animal cells. An old observation is that cytochrome C released from mitochondria during apoptosis is in the oxidized form, which predominates in vivo, whereas in the cytosol, this is rapidly converted to the reduced form by an unknown reductase or non-enzymatic reaction. Since this process is clearly exothermic, The effect of a sudden and massive release of cytochrome C into a reductive environment is in effect to provide a large increase in heat delivery to the cell, which may be crucial to the initiation of the protease cascade and to the disaggregation of the cell as a whole, being the product of an unsurvivable temperature shock. In both plant and animal cells, programmed cell death can be induced also by thermal stress, and an abrupt temperature rise has been demonstrated in plant cells undergoing at least necrotic cell death. However, the plant literature has mainly focused on heat as a cause rather than a consequence of programmed cell death. The tools that we discussed in part 2 of this series can also be used to test this idea by monitoring cytosolic temperature following the induction of apoptosis. Programmed thermogenesis is also a major part of normal physiology, with the examples shown on the slide to which we have already referred, the uncoupler protein UCP1 in mammalian brown adipose tissue and the alternative oxidase AOX in thermogenic plants. In fact, there may be many other examples in the natural world, and even rather mundane heating mechanisms that are widespread, such as the case of moths, as illustrated, which literally warm up their muscles by a cycle of contractions akin to shivering before taking flight. Warming up just like people do in the gym. Regardless of whether heat is the instrument of cell death, it's clear that mammalian cells cannot survive prolonged exposure to temperatures above 43 degrees. So this raises the question of how thermogenic tissues, such as brown adipose tissue, or the reproductive organs of thermogenic plants, can release a huge amount of heat whilst at the same time not frying themselves to death. If substantial amounts of heat are to be produced, for example, sufficient to warm up the entire body of a mouse in a frigid environment, there has to be an extremely efficient system for heat conduction through and out of the cell into the vascular system and to distant sites in the body, as well as internal protection mechanisms for the brown adipocytes themselves. Heat shock proteins, which defend against protein unfolding and aggregation, are certainly part of this self-defense. But actually delivering the heat out of the cell is a much more demanding challenge, about which we know very little. For now, all we have are rather vague hypotheses involving a cycle of heat-demanding and heat-releasing reactions. One idea, for example, is that the cytoskeleton acts as the transfer mechanism, with a heat input at the mitochondrial surface driving a conformational change in the cytoskeleton that can then be propagated to the cell surface and then released. An alternative would be conversion into a chemical form that can diffuse through the cell and then be back converted outside the cell, releasing the heat. But for now, these ideas are purely in the realm of speculation. Following the idea that cytochrome C release and its thermogenic reduction in the cytosol entrain apoptosis, we can speculate that one possible mechanism for heat transference across the cell in programmed thermogenesis posits cytochrome C as a possible heat carrier, so long as it is shielded from reduction in the cytosol and is finally exported from the cell directly into the bloodstream where it can be reduced thermogenically. A variant of this concept would have the reduced cytochrome C recaptured by endocytosis, for example, and recycled back to mitochondria to be reloaded via oxidation by cytochrome oxidase. Intracellular shielding of cytochrome C in the oxidized state would be needed to protect the cell itself from thermogenic destruction. Cell death is also crucial in immunity. There is great debate over how pathogen-infected cells and pathogens themselves are killed. Locally, there should be multiple killing strategies due to the diversity of pathogens and of their diverse strategies to survive, thrive, and overcome host defense. The delivery of packages of destructive enzymes, granzymes, is clearly one such mechanism. The granules which are transferred contain apoptotic effectors and other proteases. Cytotoxic cells, such as CD8 T cells and natural killer cells, also deliver perforins which puncture cell membranes and, at least in some cases, inducers of ROS, reactive oxygen species, which damage lipids, proteins and nucleic acids. There may also be engagement of death receptors at the surface of the target cell. Phagocytes are believed to function in a similar manner, engulfing their prey and then delivering a lethal cocktail. At this point, I'm going to hand over again to my colleague, Dr. Mugen Terzioglu, who's going to tell us a little bit about the relationship between mitochondria and immunity. Mugen.
So when we have bacterial or viral infection, there is a cascade of intricate reactions to immune system. And all these requires ATP, reactive oxygen species, or tricarboxylic acid cycle intermediates to defend the host for against the immunity. So all these related with mitochondria, and this puts mitochondria at the center stage. And now, since we have tools, we are trying to understand also the mitochondrial heat and in relation with the immunity.
The increasing evidence that mitochondria play key roles in cytotoxicity and in phagocyte activation and inflammatory signaling raises the question as to whether the unrestrained release of mitochondrial heat is also involved in killing pathogens, infected cells, and perhaps also cancer cells. Since this remains purely hypothetical for now, we can only speculate on how it may be involved. Mitochondrial products such as cytochrome C, mitochondria-derived vesicles carrying toxic cargoes or inducers of uncoupling, or even mitochondria themselves may be delivered. If mitochondrially sourced heat is a major effector of target cell death, this may explain one of the puzzles concerning fever. It was once thought that fever arises from a programmed increase in blood temperature to kill pathogenic microorganisms and viruses. However, this idea is at odds with the fact that most bacteria and viruses are far more heat resistant than our mammalian cells. This is reflected in the meltome of E. coli proteins, generally at far higher temperatures than the meltome of human cells, other than the OXPHOS proteins of mitochondria, which we've already discussed in part 2 of this series. A significant heat shock would kill the host, not the pathogen, and sometimes actually does. But if the delivery of heat from mitochondria from immune cells to infected target cells is a method of cell killing, then fever could just be a side effect of that process. Evidence has accumulated from many labs that mitochondria can indeed be transferred between cells, especially from immune cells to cancer cells. While this can also have negative consequences, it may be a common route whereby cytotoxic cells deliver a mitochondrial bomb, whether containing inducers of ROS, pro-apoptotic signals, or heat. At least 2 mechanisms for such transfer have been described: the shedding of exosomes or extracellular vesicles that are taken up by the target cell, and the elaboration of tunneling nanotubes through which mitochondria can pass directly from one cell into another. I'd like to close out this segment of our video series with a brief consideration of the possible nature of heat sensors in cells, and specifically in mitochondria, which should exist to monitor and signal when cells or organelles are overheating or are overcooled, inducing changes in respiratory activity and in coupling, as well as mitochondrial dynamics or the uptake and delivery of small molecules. These ideas are described in more detail in our 2024 Febs Journal review, so I will only give a brief mention of them here. As in many cases, the evolutionary origin of mitochondria being as once free-living bacteria, we should first consider heat-sensing systems in bacteria, such as the 2-component Des-X system that regulates membrane fluidity in response to temperature changes. Quorum-sensing ligand receptor systems are also, at least indirectly, responsive to heat. And functionally unassigned components associated with the mitochondrial dynamics machinery should also be considered, such as the SLP2 inner membrane organizing protein. In the next part of this series, we'll be looking at some major evolutionary issues, both ancient and more recent, that I believe may be partly addressed by consideration of mitochondrial heat production. We'll start by posing the question of whether aerobic bacteria are also hot, like their mitochondrial descendants, then discuss the role heat may have played in the emergence of eukaryotes and later of homeotherms. I believe that OXPHOS today plays a role in keeping organisms warm that was once purely performed by geothermal processes. I'd like once again to thank my funders and colleagues, especially Mugen Tessiolo, Martin Sibor, Pierre Rustin, Paul Bény, Oriane Shirihai, and Eric Dufour, who have co-developed the ideas put forward here and pointed me in the direction of relevant literature. Thanks also to all image originators and sources used here.